Assalamualaikum and hi, I am Nur Ainuna Jua binti Muhammad Fazli, metric number 202648. Today, we are going to do two experiments, which are streaking for single isolated colonies and gram staining. The principle of streaking for single isolated colonies is this technique commonly used for isolation of discrete colonies, which initially require that the number of organisms in the inoculum be reduced. The resulting diminution of the population size ensures that the individual cells will be sufficiently far apart on the surface of the agar medium to separate the different species. The streak plate method is a rapid qualitative isolation method. It is essentially a dilution technique that involves spreading a loopful of culture over the surface of an agar plate. Okay, let's start our experiment. These are the materials and equipments that we're going to use today. This is our agar plate, our bacterial culture, inoculating loop, permanent marker, and Bunsen burner. Firstly, turn our agar plate upside down and don't forget to write our bacterial culture name, then our name, and lastly, the date of our experiment. Done. Okay, then hit the loop and make sure to cool it first. Then, take the bacterial culture and immediately streak to the agar plate in the quadrant 1. Then, hit again the loop and make sure to cool it first. Turn the plate 90 degrees, touch the corner of quadrant 1 and drag it to quadrant 2. Reframe the loop and make sure to make it cool first. Then turn the plate 90 degrees, touch the corner of quadrant 2 and drag it to quadrant 3. Without reframe, drag the quadrant 3 to the quadrant 4 and streak it. Reframe the loop and let it be cool. And we are done! These are the expected result of isolated mixture culture between S. mercescens and S. aureus. These are the following description for the colony form. While this is the expected result for the isolated mixture culture of E. coli and S. aureus. And this is the following description of colony form. The principle of gram stain is it requires the use of at least 4 chemical reagents. The first reagent is primary stain, which is crystal violet. Its function is to impart its color to all cells. The second stain is a mordant, which is iodine. It is used to intensify the color of the primary stain. To establish a color contrast, the third reagent, which is ethyl alcohol 95%, act as decolorizing agent. This decolorizing agent may or may not remove the primary stain from the entire cell or only from the certain cell structures. The final reagent is the counterstain, which is safranin, that has a contrasting color to the primary stain. Following decolorization, if the primary stain is not washed out, the counterstain cannot be absorbed and the cell will retain the color of primary stain. While if the primary stain is removed, the decolorized cellular components will accept and assume the contrasting color of counterstain, so the cell can be distinguished. Okay, let's begin our experiment. The materials and equipments that we need are crystal violet, tap water, iodine, ethyl alcohol 95%, safranin, fibrous paper, dropper, broad culture, and inoculating loop. Firstly, heat the loop and make sure to let it cool first. And then open the cap of the broad culture bottle and heat it first before we take the bacterial culture. Then, smear our bacterial culture onto the prepared slide. Then, heat fixation is performed to avoid the bacterial smear from being washed away during the staining procedure. Next, take our crystal violet and then gently float smears with crystal violet and let it stand for 1 minute. Next, gently wash it with the tap water. Then, float smears it with the grams iodine mordant and let it stand for 1 minute. Gently wash with tap water. Then, decolorize it with 95% ethyl alcohol. Make sure to do not over-decolorize it by adding the reagent drop by drop until the alcohol runs almost clear. Gently wash with tap water. Then, counterstain it with safranin for 45 seconds. Gently wash it with tap water. And lastly, blot dry with Bibulus pepper. And we are done! Gram-positive cells have a thick peptidoglycan layer, while the gram-negative cells have thinner peptidoglycan layer and surrounded by outer lipid-containing layers. So, for the expected results, the gram-positive bacterial cells have a distinctive purple appearance when observed under a light microscope. This is due to the retention of the purple crystal violet stain in the thick peptidoglycan layer of the cell wall, while the gram-negative bacterial cells, which have a thinner peptidoglycan layer, will allow the crystal violet to be washed out. They will stain in pink or red by the counterstain, which is safranin. Okay, that's all for today. Thank you.